In the next 25 minutes, I will be presenting some of the results on the validity of the digital mobility outcomes that were obtained in the clinical validation study of Mobilise-D. I won't show all the results, just pieces of it. The technical validation study provided us a set of algorithms that allow the calculation of digital mobility outcomes at the walking bout level. This means that for every walking bout of a person, so this is a period of consecutive steps preceded and followed by a resting period, we have information on the duration, number of strides, walking speed, cadence, stride and step duration, stride and step length, and number of turns. This is the original list of the DMOs that we have at a walking bout. And these DMOs have been proven valid when they were tested for criterion validity in the technical analysis. So let's say we have walking speed of one person in one walking bout. Is this clinically valid? Or is this walking speed clinically valid, or this one? So actually, we underwent all the procedures, the traditional procedures to approach clinical validity. So we did construct validity, predictive capacity, and ability to detect change. But because of the complexity of the data and the novelty of the approach, we had to use also additional analysis before we were able to test the clinical validity. The first challenge was to go from the bout level to the real world. And this is the work that we call the aggregation analysis. As I said, we have information on the DMOs for every walking bout of a person. And because the patients participating in the study recorded 24 hours of 7 days, we have hundreds or thousands or 100,000 of bouts per person, which means that for every person we have thousands of values for every DMO. And we want, so let's say, what's the walking speed of a person? We need to find one single value per individual. And we want this single value to represent real life as much as possible and to be clinically meaningful. We searched in the literature about digital devices and digital data, and despite the use of digital devices, we didn't find any framework or anybody who said how we can aggregate. So we aimed to first develop an aggregation framework that is parameter agnostic. Let's say we used it for mobility, but anybody in the future could be using this for body temperature, oxygen saturation, and then we applied this framework to our digital mobility outcomes. In the left part of this slide, you have a very reduced version of our framework. It has 3 parts that are sequential, and one of these parts can be iterative depending on the temporal levels. In all of these steps, we combine statistics with experts' discussion, and experts means clinical experts, technical experts, and patients' input. We defined that the aggregation is a combination of what we call restrictions and summary statistics. So let's go to the walking speed example. What is the walking speed of a person in the real life? It could be the mean of the walking speed as we measure in every single bout of these thousands of bouts, or it can be the median, or maybe the best clinical or the most informative summary is the maximum walking speed. So this would be the summary statistics. But then we can calculate the mean of the walking speed in all bouts, or the mean of the walking speed in the bouts that are, let's say, longer than 30 seconds, because we may consider that the shorter bouts are not that clinically relevant. And this is what we call restrictions. So when we have a list of aggregations per each DMO, and is this the combination of restrictions and summary statistics that makes sense, we want to evaluate which one is better. And because we want to take the most out of every single DMO, we wanted to select the aggregation method that provides the highest stability result in the DMO and the most ability to discriminate in the DMO. The stability, because we understand that if something is stable, it's more representative of real life, and the ability to discriminate because it brings clinically meaning. We tested the first with intra-class correlation coefficients and the second with the area under the curve comparing disease with healthy people. This slide shows one DMO, how we present the results. So for each DMO, in this case that's walking duration, we tested different aggregation methods. In this case, that's walking duration, so the summary statistic was the sum. What makes sense for walking duration is during the day to sum all the durations of every bout, to have walking time in a day. And then for several restrictions, so for all bouts or bouts longer than 10 seconds, 30 seconds, 60 seconds, etc. We calculated ICCs and AUC values for each of the diseases. These two estimates range from 0 to 1 being the darker and meaning better stability and better ability to discriminate. Then we did this for all aggregations and for all of the walking bouts. In general, you can see that the ICC is darker, so we obtained quite high or very high ICC. This means stability of the DMOs in the different aggregation methods for most diseases and for most DMOs, with some exceptions. Let's say this, for example. But then in terms of the area under the curve, so this is the ability to discriminate between healthy and disease, the results were much more heterogeneous, and there were differences by DMO and differences by disease. When we observed this, we took the decision to select more than one aggregation method for some of the DMOs, because they may bring different clinical information. So at the end, from the original 15 DMOs that the technical validation study provided us, we ended up with a list of 25 DMOs at the week level that we hope represents real world. These DMOs are classified in two big groups, walking activity and gait DMOs. Within walking activity, we have volume DMOs, walking duration, number of steps, number of turns, and pattern DMOs, number of bouts of different durations, bout duration, maximum duration, and walking bout duration variability across bouts. In the gait DMOs, we identify pace, rhythm, and bout-to-bout variability. In pace, we have walking speed, maximum walking speed, and stride length, and we have them calculated for different restrictions of the bouts. For rhythm, we have cadence and the stride duration, again, average values and maximum and in different duration of bouts. And for bout-to-bout variability, walking speed variability, stride length variability, cadence variability, and stride duration variability. We anticipated when we did this work that some DMOs maybe work across diseases, but some DMOs will be disease-specific, and that is okay. This is what we are looking for. In the next slides, I will be showing results, not every result for every DMO for every disease. I will select just some pieces of it for simplicity. A second part in this, going from the bout to the real world, is to define what is the minimum wearing time of the digital mobility assessment that is enough. So this one single value that we have summarized from all the bouts, we want it to be valid and precise for each of the DMOs and representative of real world. And then we operationalize this in three different objectives. We want to identify the minimal wearing time in a day that constitutes a valid measurement of the DMO, the minimal number of days that constitute a reliable weekly aggregate, and the number of weekend days that are required to get a reliable aggregate of the DMOs. So again, we had for each DMO and each of the diseases, we obtained a minimum number of hours per day that would be required, a minimum number of days per week that would be required, and a minimum number of weekend days. And when we have this for all the DMOs, and I'm not going to read it, in general, we observe that there are no relevant differences between diseases. There are differences between the groups of DMOs. So some DMOs are more sensitive, let's say, to the wearing time. And then we went on a very conservative approach. So we want to select the minimum time that would allow most of the DMOs to be valid and reliable. And we ended with the decision of defining a valid and reliable mobility assessment as at least 12 hours of wearing time during waking hours, during 3 or more days in a week without weekend requirements. Then, once we have one value per DMO and per patient and visit, and before or in addition of testing the validity, we need to test whether this digital mobility assessment is feasible at all. And here we have very good news, I think. In the clinical validation study, of all the patients we included, 94% wore the device, 94%. And 90% of all the sample wore the device the minimum time required, so had valid and reliable assessment. Among them, they adhered to the protocol optimally. The average wearing time was 6.8 days, 22.8 hours. They were asked to wear this during 7 days, 24 hours. So this is virtually perfect. More important, we tested whether there are differences between the wearers and non-wearers, so those who were wearing the device and those who didn't. We found no differences in age, gender, smoking, living status, or mental health, comorbidities, functional capacity, health status, or even history of falls. We found a difference. It is slight, but it was statistically significant in BMI. Those who wore the device had slightly higher values of BMI than those who didn't, and a difference in the proportion of people with frailty. And actually, of the several components of frailty, the one that was different was BMI. As you know, this is one of the components. Okay, then, before we go to testing, there is also another step that is kind of more intuitive. If we want the DMOs to be tested for clinical validity, they need to be computable and they need to be informative of something. So we first described the DMO's distribution in each of the diseases, and we want them to be discriminating, to say, let's see, to be distinguishing healthy versus diseased, because we know that these diseases have mobility impairment. We want them to have variability and to have a distribution that is plausible or according to our knowledge. I'm showing here the results for number of steps, one of the volume DMOs in COPD. We found that people with COPD have lower number of steps than healthy adults. This is something that we know from the literature, from many other, or from different tools, right? And the fact that we find it here is a proof that these DMOs are measuring what we want them to measure. We have also found a difference in speed, real-world walking speed in COPD in comparison to healthy adults. This was not known, but it makes sense. And we didn't find a difference between stride duration, that's a rhythm DMO, between COPD and healthy adults. And this also makes sense because we wouldn't expect this DMO to be affected by a chronic obstructive pulmonary disease. So we have the DMOs that are able to distinguish the disease from healthy, but still the DMOs exhibit variability. And this is also important because it's been mentioned in the morning that we are talking about performance, not about capacity. And we know that out there is variability. So in comparison to, let's say, all the patients having the same value of steps per day, we have a very wide range of steps per day. So in this case, the COPD patients walk from a few hundreds to more than 20,000 steps per day. And this kind of normal skew to the right distribution matches what we know from the literature using other devices. When we test the distribution of other DMOs that are not known in COPD, and again, maximum walking speed and cadence, they also have a wide distribution, and this is suggesting that the DMOs have potential to be used for disease characterization. So they are giving some information that maybe the traditional measures used in COPD do not give. And we are working on identifying this. And now I'm moving to the traditional approach to validity of the DMOs. So first, construct validity. What we wanted to prove in the construct validity concept means that what you are measuring is assessing exactly what you want it to measure. And you test it with different properties. One of them is convergent validity, the ability to which the DMOs relate to constructs that we expect them to relate. Divergent validity, the ability of the DMOs to not relate to constructs that we expect not to be related to. And known groups validity, which is the ability of the DMOs to distribute differently across subgroups of the population that make clinical sense. Very important in this construct validity analysis is to define a priori hypothesis. So we had to work the statistics and the technical teams with the clinical experts to define what we expect to have convergent validity, divergent, and known groups. And then the assessment of construct validity is not about whether the correlations are high or are low, but whether they match what we expected. This table shows the convergent validity results of the walking activity DMOs in multiple sclerosis. We tested the correlation with 3 clinically relevant measures in this disease, and for each of these combinations of DMOs and clinical measure, we had a range of the expected correlation, let's say from 0.5 to 0.7. Then here we painted in green the correlations that match our a priori hypothesis. Overall, the convergent validity of these parameters is quite good. There is one parameter that doesn't show good convergent validity, so maybe this is not a good DMO for this disease. This table shows divergent validity of the gate DMOs in Parkinson's disease. Again, we find that all the DMOs have very good divergent validity, and actually this is happening for all DMOs and for all diseases. And here you see the results of known groups validity for some DMOs in Parkinson's disease. In this case, the hypothesis as put by the clinicians was that the DMOs in Hoehn and Yahr stages 1 and 2 would not be different, and they would be different from 1 to 3 and from 2 to 3. This is one of the PACE DMOs, maximum walking speed, and as expected, there are no differences between Hoehn and Yahr 1 and 2, and Hoehn and Yahr 1 and 2 have a different value than Hoehn and Yahr 3, as expected. We see a very similar or the same pattern in walking speed variability, so this is one of the variability DMOs. However, we do not see this with this pattern DMO, walking bout duration variability, which suggests that this DMO doesn't have good known groups validity. Then we have also tested predictive capacity of the DMOs. Predictive capacity is the ability of the parameter to forecast a future outcome. And in this case, we want to evaluate 2 things. One is the magnitude of this predictive capacity as measured with the area under the curve. This is a parameter that usually ranges from 0.5 to 1. The higher, the better. So the higher means that the DMO is able to predict perfectly the outcome, in this case, death. So we want them to be to the right, but we also compare the DMOs with known predictors. In this case, maximum walking speed, it's moderate to good predictor of mortality, but it's actually better predictor of mortality than age. So we have done this with all DMOs and for some of the outcomes. And in this figure, I'm going to guide you through the prediction of mortality. During the first 12 months of the project, we had 3% of people who died. Some of the DMOs, and these are the walking activity, volume and pattern, and the PACE DMOs, have moderate to good ability to predict death. More importantly, they have higher ability to predict death than the most accepted mortality predictors. This is age, quality of life, comorbidities, frailty, function, exercise capacity, loneliness, or cognitive function. Not all the DMOs work the same way. In this case, rhythm and variability DMOs show poor to moderate ability to predict death, although it's slightly better than the ability to predict death of the most accepted mortality predictors. I'm jumping 3 slides now because I want you to go back to the very first slide where Lynn was saying, is mobility the 6th vital sign? So according to these results, it may be. We have tested other outcomes, and I'm showing just one example. This is the improvement in activities of daily living after a hip fracture. And apologies, because this is 6 months improvement, not 12 months. 10% of the patients with hip fracture improved their ADLs during this period. And again, we observed that the walking activity and gait DMOs have good ability to predict this improvement, which is much better than the ability of many known predictors. So finally, ability to detect change. We want the DMOs to be able to detect change when change happens. So what we have done here is to test whether the DMOs change at all, whether the patients experience changes, and whether these 2 changes are related. In this figure, you see how 3 DMOs, one for volume, one for pace, and one for rhythm, have changed over time in COPD. That's baseline, 6 months, and 12 months. So in general, they have declined. We asked the patients at the 12 months visit, compared to your first visit, how was your walking? And they say it has improved or it has worsened. So much better, a little better, the same, a little worse, much worse. In this figure, we see the distribution of the changes in walking duration according to these categories, from much better to much worse. And we observe that in patients who report that their walking has been better during these 12 months, walking duration actually increased. And in the patients that reported that walking was worse, walking duration had decreased. And there is a kind of dose response here. And this also was observed for other DMOs. So this is walking speed and cadence. So I'm going to summarize. We have seen with this first analysis, as I said, these are early insights we have not finalized. We see that digital mobility assessment is feasible with very good adherence to the protocol, and this is actually good results if we want to deploy this or to use these tools for monitoring chronic diseases. And that's been nicely presented by Clemens, the need of doing this. We have found that these DMOs have potential to be able to provide additional information to that of the clinical measures in people with chronic diseases. We found overall quite good construct validity, but there are differences between DMOs, and there are differences in which DMOs have better construct validity according to disease. So we need to work a bit more on this. Some of the DMOs have good predictive capacity, but others don't, and some outcomes can be predictors and others not. I've not shown everything, but we're working on this. And some of the DMOs, again, are able to identify change when the change happens. And I think that's very important if we are thinking in, let's say, using the DMOs as outcomes for clinical trials. So the next steps, we need to finalize the analysis. We just got the data very recently, so we need to do everything to interpret properly. At the midterm, what we want is to be able to obtain from the 25 candidate DMOs that I listed, the set of DMOs that is valid and useful and clinically meaningful for each of the diseases, and if which would be the DMOs that can work across all of the diseases. We selected these diseases with different mobility disability patterns or causes, and if we identify something that works well for all of them, this could eventually be used for any other chronic disease. Thank you.